Today I want to talk about stomata. And in particular, stomatal responses to environmental conditions. So what do guard cells respond to? What environmental conditions do guard cells respond to? Yeah, that's a good one. Although exactly what that response entails is kind of up in the air. No, I didn't mean to. Humidity or vapor pressure, or how dry the air is, or how quickly the transpiration rate, how high, what the rate of transpiration. That's definitely it. So something related to the dryness of the air, or how quickly water is being lost. That's one. What else do stomata respond to?
CO2 concentration.
CO2 concentration. That's good. That's a good one. Not all stomata respond to that, but many plants do, and some plants respond to CO2 only when they're water stressed, but not when they have plenty of water. Some plants don't respond to CO2, but in general, yeah, CO2 is a good one. Those are two things now. And I'm going to spend some time talking about each of these in turn, right? More time than just waving my hands. What else?
Possible light.
Light. That's arguably the most important environmental factor to which stomata respond, is light. And it's a rather complicated response, but probably the most is known about that response of guard cells to light than any other condition. So, what did we say? Humidity, or vapor pressure or something? CO2 and light. What else?
About the solids, the guard cells, and
So that's more like the mechanism whereby they respond, yeah, and it has solutes, but what about like the environment? What about what's outside the plant that they respond to? Soil water content, that's a great one, yeah. That one's important, very important. And the way they do that probably is through the hormone abscisic acid. And so there's a response of guard cells to abscisic acid, and that response is tied into drying soil and things like that. So that's an important one too. So those four are the ones that I want to talk about in some detail. Those four environmental conditions. How dry the air is, how dry the soil is, and abscisic acid, light, and CO2. I can't think of any others right now, but maybe there's something I'm forgetting. Notice that I didn't say leaf water potential. So what I'd like to do is talk about those in more detail and start with light, arguably the most important environmental factor to which the model responds. Light. And so I showed this picture before. By the way, I have your exams graded. I'll pass them out towards the end of the period. I'd like to spend today just talking about light response of stomata. So I don't know how long it's going to take, but maybe towards the end of the period, I'll just pass these out. Okay. You saw this picture before. So here's stomatal conductance, and here's light intensity. This is photosynthetic photon flux density, in case you're not familiar with this abbreviation. Photosynthetic photons, that's in the wave band 400 to 700 nanometers. Photons in that wave band. This is micromoles of photons per square meter per second. So this is higher light, lower light. Although, what's the photosynthetic photon flux density for full sunlight? What number is that? Does anybody know? It's about 2,000 micromoles per square meter per second. This is only going up to 400. So this is still not even half of full sunlight. It's kind of dim condition. By the way, you know, your eye isn't a good sensor for light. You can't really tell how bright it is by trusting your eyeballs, because they compensate. They make everything look bright, even when it's not so bright. Right? They're really good sense. They're really good in that sense. You know, they keep things constant. But to use it for an instrument, no, your eye isn't very good. Anyway, I mean, it looks pretty bright in here, but I bet if we had a quantum sensor, we'd be way below 400 micromoles per square meter per second of light in this room. Anyway, it's not very bright light, but you notice that at lower light, the stomatal conductance is lower, and as the light increases, the stomatal conductance increases. So the stomata open in response to light. And it doesn't take very much light, especially in this species, to saturate that response. This is by about 100 micromoles. The conductance is saturated. You know, it's not going to increase anymore. This maximum stomatal conductance and the nature of this curve, where it saturates and stuff, depends on the environmental history of the plant, as well as what species it is and what's been happening. So this is for a conifer in South America, but here are some light responses for oak trees in New Jersey. And this shows, again, here's light intensity, and here's the stomatal conductance. And this one does go up to full sunlight. And you can see in these three species that their maximum stomatal conductance is much higher before they imposed a drought versus after they imposed a drought, or after a drought happened or something in New Jersey. And these are the three species. Also, let's take a look. This maximum conductance is about 0.1 mole per square meter per second. These conductances, this is millimoles, so here's 0.1 mole. So before the drought, these had a higher conductance than that last tree, but after the drought, they all had about the same level. So the nature of this light response depends on the environmental history of the plant and what it's experiencing, as well as what species it is, all kinds of other things. But in general, the general response is stomata close in the dark and open in the light. There are two separate systems that guard cells have to sense light. And these are called, for whatever reason, a red light response and a blue light response to stomata open. And these are two separate systems, two separate things that make the guard cells respond. Okay, so for historical reasons or for whatever reasons, they're called red light and blue light. The blue light response is, in fact, a response to light in the blue part of the spectrum. The red light response is a general response to light, any kind of light, red, blue, green, whatever. But it's called red. Okay, so let's go into some evidence for these two different systems to respond to light. If you look at the maximum stomatal conductance, this value here, when light is saturating, that maximum conductance, it's higher in blue light than it is in red light. So you can shine red light on a leaf and the stomata will open. And as you increase the light intensity, they'll open more and more and more until you saturate, until you get to some maximum where increasing the light won't increase the conductance anymore. But then if you add a little bit of blue light to that, they'll open up further. That's weird. Okay. That's some evidence for two separate systems. Also, blue light alters the red light response in a non-additive fashion. I'll go into these in some detail on the next few slides. So again, if you have, well, you'll see in the next few slides what I mean by non-additive. Another bit of evidence is this chemical, dichlorophenol dimethylurea, DCMU. It blocks photosynthetic electron transport in the Z scheme of photosynthesis. This chemical eliminates the response of stomata to red light, but it doesn't affect the stomatal response to blue light. This is it's an herbicide called diuron. And here's this confocal image of the two guard cells with chloroplasts in them. And microtubes in green, right? Look at these things. You know, chloroplasts move inside the cell, back and forth, and they change their position inside the cell. You know, it's weird. That's so weird. It's like a alien. Okay, so let's go into this. Here's a article that shows that stomata open further in blue light than red light. So here we have the stomatal conductance on this axis. Again, here's that number again, 0.1 moles per square meter per second. And this is time over here, 30 minutes, like this. And what they did was they had a red light at 500 micromoles per square meter per second, so probably saturating red light, amount of red light, and they got this much stomatal conductance out of that. Then they switched on a dim blue light, 25 micromoles, not very bright. And when they switched it on, the stomata opened way up further. It took them a while to do it, right? This is a 30 minutes of 1 to, hour and a half or something to respond. But they opened way up. And it didn't take much blue light to do it. If you would have come along here and given them 25 micromoles more red light, you wouldn't see a response. But the blue light makes them open up. So there's something about blue light that's different than red light. And this is for this plant here, online. Okay, so, what was my thing? Maximum stomatal conductance is lower in red light than blue light. Okay, so it takes some blue light to really get the stomata wide open. Okay. Now, this next one is blue light alters the red light response of stomata in a non-additive fashion. So here we go. Look at this. This is even stranger. Here's now this is again, it's time on this axis. And here we have a change in conductance. So they did things, and then you can see how that affected the conductance, what the change was. So in this first trace, their conductance was constant at whatever it was, 500 micromoles per square meter per second of red light. And then they gave it a pulse of blue light, 50-second pulse, less than a minute. Okay, so here's 15 minutes. So this pulse is turn on the blue light here and turn it off there. And look what happens, the conductance increases. You get a change in conductance due to that pulse of blue light. Even though you turn the light back off again, still the stomata open up. And then eventually they go back to where they were before. That's what I mean by blue light alters the red light response in a non-additive fashion. A brief pulse elicits a long-term response. So again, there's something different about blue light than about red light in the responses. Stomata. Here they gave it 2 pulses, and what was C is, same thing. Same idea, that there's something inherently different about blue light and how guard cells perceive that blue light than the way they perceive red light. Here's the DCMU one, okay? So here, this is the herbicide DCMU diuron. And here's the electron. I got this slide today because I didn't want to have to draw it on the board again like I normally do and get all messed up. Anyway, this is these are the light reactions of photosynthesis. The Z scheme, the photosynthetic electron transport that's happening in the thylakoid membrane in the chloroplast. Okay, so here's photosystem 2, photosystem 1. And normally the electron comes from water, goes through this chlorophyll, comes down here through these acceptors, plastoquinone, cytochrome, plastocyanin to the cellular chlorophyll in photosystem 1. It gets excited, it transfers its electron here, finally to make NADPH out of NADP. The resulting electron transport moves protons from one side of the thylakoid membrane to the other, and then that proton and pH gradient in the membrane is used to synthesize ATP from ADP and inorganic phosphate. So the end result of this is ATP plus NADPH. Those 2 chemicals go on in a Calvin cycle to fix it up. DCMU blocks this electron transport right here. Doesn't let it happen. And it actually competes with plastoquinone at one of the spots, and it's a complicated thing. If you want to know some of the details about it, I recommend this book as a starting point, not physiology. But anyway, it blocks electron transport here, so that the plant can't make NADPH, it can't make ATP through photophosphorylation. And eventually, it'll kill the plant, right? It's an herbicide. DCMU inhibits the red light response of photosynthesis. It eliminates that response. They'll still respond to blue light, but in the presence of DCMU, guard cells don't respond to red light. So again, there's something different about those 2 light responses. Red, blue. Paraquat is another herbicide that affects electron transport. If that paraquat siphons off electrons up here in this part, then it makes superoxide that bleaches the leaf out and kills the plant that way. But anyway. So there's a class about that right here. You want to know about herbicide mode of action? Do they still teach that course?
Yeah.
Is that, who teaches it? John Lindquist?
Lindquist, yep.
Yeah, okay. Yeah. So if you want to know more about that, there's a whole course about how herbicides work. And a few of them are photosynthetic inhibitors, like diuron and paraquat. What's the big one? Roundup. That doesn't work this way. Roundup works a different way. And so does the nasty one, dicamba. That works. Have you taken the course?
Yeah. I took it in undergrad.
Oh, okay. So you could tell all about that. How does dicamba work? Do you remember?
I used to know all the modes of action, but kind of faded away.
I used to know that. I know I heard it before, but I just can't remember it. Anyway, it doesn't work this way. And in fact, when people were doing research to try to figure out what all these compounds were in the electron transport scheme, chemicals like this were quite useful in blocking the electron transport at different points, and then you can use that info to figure out what's what, you know, what the redox potential is of something to do whatever it is. So anyway, it's not just an herbicide, it can be a useful tool as well. Okay, so DCMU blocks, it inhibits, it eliminates the red light response of stomata, but not the blue light response. That's it, I'll come back to that. Because the question that arises then is one that came up the other day: how does this work? How do guard cells sense that light? What's the photoreceptor? What's the sensor in the cell that lets the cell know that the light's turned on? Right? Okay, now remember, I meant to do this at the beginning, but remember remember the sequence of steps that happens when a closed stomata opens. So what happens in the guard cell? The first thing is it gets a signal, some signal, like the light turns on. What does the guard cell do? It starts pumping protons from the inside of the cell out into the apoplast. That sets up a gradient across the plasma membrane in pH and electrical potential. That's important in allowing potassium ions, positively charged potassium ions, to come from the apoplast into the guard cell. Because the positive charge, the guard cell is pumping positive charge out. So that positively charged potassium wants to come back in like a magnet, right? So potassium comes in. Chloride ion comes in, as we said before, and that may be an active process because that's moving against its electrical gradient. But increase in potassium, increase in chloride concentrations, and then also increase in malate concentration, the organic ion malate. And that may be a response in trying to maintain a constant pH, as I talked about last time. And then that change in osmotic potential, because of the accumulation of potassium and chloride and malate, decreases the osmotic potential, makes the guard cell not in equilibrium with the total water potential outside of it. Water moves across the membrane into the cell, increasing its volume, distorting the cell, and opening up that pore in between the two guard cells. So the first thing that happens is protons get pumped out of the cell. But what is it that senses that the light's turned on and tells this guard cell, okay, start pumping protons out there? Well, it's got to be a couple of things. You have to have a sensor to tell when the light's on, and then you have to have a source of energy for pumping those protons out. So I guess there's two questions. How do you sense it, and how do you get the energy to start that proton pumping that sets everything in motion and opens the guard cell? Well, for the red light response of stomata, the photoreceptor is chlorophyll. The molecule that senses when the light's turned on is chlorophyll. That absorbs photons from that light is chlorophyll. So here's the absorbance of chlorophyll A, chlorophyll B in vitro. And there's just a tiny difference between chlorophyll A and chlorophyll B, but both of these are found in vivo. And you see the absorption spectrum here. Chlorophyll is the photoreceptor for the red light response of stomata. Now notice, when I say red light, that's this part of the spectrum. But chlorophyll also absorbs here in blue. And in vivo, it also absorbs a fair amount in the green part of the spectrum too. And so, as I keep saying, red light response, it's a general response to light. It's not specific to red light. The blue light response is specific for bluish light, but the red light response is across the whole photosynthetic reactive spectrum. But anyway, photoreceptor is chlorophyll. How do we know that? Well, DCMU closes the stomata in the presence of red light. So you got open stomata, you can start in the dark, turn the red light on, or turn the light on, stomata open. Put DCMU there, and the stomata close. Not only is the photoreceptor chlorophyll, but the energy to pump protons out into the apoplast comes from photophosphorylation, right? Comes from production of ATP through photosynthetic electron transport in the Z scheme in the chloroplasts of the guard cells. All right, so here's a simple Z scheme right here. Right? Of electron transport. Here's where DCMU blocks here, paraquat blocks over here, and that. But this, in the normal transport of electrons this way, we make NADPH, and we make a lot of ATP. That production of ATP by this scheme is called photophosphorylation, and that's a source of energy. That ATP that's produced by photophosphorylation can be used to pump a proton out into the apoplast across the plasma membrane. Because proton doesn't go there by itself. It needs help. It needs energy to move across the membrane. And that energy in the red light response of stomatal conductance comes from photophosphorylation. How do we know it? Well, DCMU inhibits it, and it makes open stomata closed, even though the red light's turned on. There's more evidence than just that. Okay, between 600 and 700 nanometers, okay, so in this red part of the spectrum over here, the photosynthetically active light is also the light that your eye is sensitive to. So we can see this as red. We can see this light down here as blue. But in this red part of the spectrum over here, if you make an action spectrum of stomatal open, and I'll get into that next slide. You make an action spectrum of stomatal open. It's virtually identical with the action spectrum of photosynthetic carbon fixation. What is an action spectrum? Remember this? Okay, it's nothing more or less than some action, and in here, oxygen evolution rate is plotted on the y-axis. That's the action. And then you take monochromatic light of whatever wavelength and shine it on your organism and measure this action and get a response. This dashed line is the action spectrum of oxygen evolution. And you know that oxygen, remember, oxygen is produced when this electron comes from water. To start this whole process, we have to end up with oxygen. This oxygen evolution is a measure of the photosynthetic rate. Can be used to measure photosynthetic rate. Usually not used to measure photosynthetic rate anymore because we have so much oxygen around all the time anyway. So, you have 20% oxygen in the atmosphere. How much more is your little leaf going to add to that concentration? Not very much. But if you look at CO2 concentration, where there's only parts per million of CO2, then the decrease in CO2 is easier to measure. So oxygen evolution historically was a way to measure photosynthesis, but not used very much anymore. But that's digression. We can think of this as a photosynthetic rate. That's this. If you look at the absorption spectrum for, this is intact chloroplasts, you see that they match quite well, especially in this red part of the spectrum. That's evidence that the absorption of light by whatever is causing this action. So the absorption so for this picture, the absorption of light by intact chloroplasts is responsible for photosynthesis. I mean, that's something that we all know. That's old news for us. But we could have an action spectrum of stomatal conductance and an absorption spectrum of chlorophyll. And if you plot those, again, you get the same in the red part. You get an overlap of those two curves, which indicates that absorption of light by chlorophyll has something to do, or it provides evidence that the absorption of light by chlorophyll is responsible for stomatal opening. That's an action. Okay, so going back, in the red light region, 600 to 700 nanometer light, the action spectrum of stomatal opening is virtually identical with action spectrum of photosynthetic carbon fixation. I tried to find a nice graph that showed it explicitly, not so easy. I'm still looking. So if anybody comes across one, let me know. I mean, you see statements like this all over the place. Look, here. But they don't give the action spectrum. It's complicated. Okay. All right. And before I leave this slide, I was wondering why I put it on, but in the blue light response of stomatal opening, just like in the red light response, the first thing that happens is protons get pumped out of the guard cells. Just keep that in mind. This will come back to that later. Okay. So these action spectra of chlorophyll absorbance lines up with the action spectrum of stomatal openings lines up with the absorption spectrum of chlorophyll in this red part of the wavelength. Evidence that chlorophyll is a culprit. Here's another one. This one's very interesting. There's a genus of orchids called Paphiopedilum. You know that? Does anybody grow orchids? Some of you? George Graff grows orchids. You know George Graff's maybe read.